The overall goal of this neurophysiological experiment is to assess changes in transmission at the corticomotoneuronal synapses in humans after repetitive transcranial magnetic stimulation.
The age-reflex conditioning technique can help answer key questions in the field of neuroscience, such as better to understand basic mechanisms and sites of neuroplasticity. We were especially interested in the plasticity taking place between the corticospinal tract and the motor neuron, the corticomotoneural synapse.
The main advantage of this technique compared to measuring compound potentials is that it is possible to measure synaptic input produced by distinct corticospinal connections to spinal motor neurons.
There are at least two further advantages of conditioned age reflexes compared to compound potentials. First, it's possible to detect influences of even weak corticospinal volleys. And second, it's possible to assess effects of longer latency corticospinal volleys.
Prior to the experiment, first screen the subject for TMS medical risks such as history of epileptic seizures, obtain written and informed consent, and inform the subject about the purpose of the experiment. Prepare the skin for surface electrode placement over the M. soleus muscle by shaving, disinfecting with propanol, and applying light skin abrasion. Then, place surface electrodes to measure electrophysiological responses. Check the EMG signals on the screen while the subject performs plantar and dorsiflexion.